Welcome to G Fun Facts Online. Today, we're diving into a silent war that has raged for millennia, a conflict unseen by the naked eye, yet one that has shaped the very fabric of life on Earth. This isn't a battle fought with tooth and claw, but with molecules and mimicry. It's the clandestine struggle between hosts and their most intimate adversaries, parasites. These invaders, from single-celled protozoa to complex worms, have evolved an astonishing array of strategies to outwit, subvert, and manipulate our sophisticated defense systems. This is the story of their biological ingenuity, a tale of espionage and sabotage at the cellular level. Imagine trying to fight an enemy who changes their uniform every few minutes. By the time you've identified them, they look completely different. This is the core strategy for many successful parasites, a tactic known as antigenic variation. It's one of the most dramatic evasion strategies in biology. The parasite constantly changes its surface coat, the very thing our immune system learns to recognize. It's like a never-ending masquerade ball, and our immune system is always one step behind. The quintessential master of this disguise is the African trypanosome, Trypanosoma brucei, the parasite that causes sleeping sickness. The surface of this parasite is covered by a dense coat of about 10 million identical protein molecules called variant surface glycoproteins, or VSGs. Think of it as a single, uniform armor. But here's the trick. The trypanosome's genome contains a massive library of over 1,000 different genes for these VSG proteins. At any given moment, it only wears one type of armor. But just as the host's immune system finally produces the right antibodies to target that specific armor, the parasite switches. It activates a new gene from its library, shedding its old coat and replacing it with a brand new, unrecognized one. This switch is a feat of genetic engineering involving complex DNA rearrangements. The result is a cycle of rising and falling parasite numbers in the blood, causing the characteristic waves of fever and sleeping sickness, as our body constantly plays catch-up. But the trypanosome isn't alone. The malaria parasite, Plasmodium falciparum, pulls off a similar masquerade. After infecting a red blood cell, which normally has no surface markers to alert the immune system, the parasite decorates the outside of the cell with its own proteins. The key protein here is called PFEMP1. This protein is not just for show. It's a virulence factor that makes the infected red blood cell sticky, causing it to cling to the walls of blood vessels. This prevents the cell from being swept away to the spleen, which acts as the body's filter for old or infected red blood cells. Just like the trypanosome, the malaria parasite has a family of genes for these sticky proteins, about 60 of them, called VAR genes. And again, only one VAR gene is expressed at a time. The switching process here is controlled epigenetically, meaning it's not the DNA sequence itself that changes, but how the DNA is packaged. Silent genes are kept tightly wound and inaccessible. To switch, the parasite unwinds a new gene, making it readable while silencing the old one. This constant antigenic switching allows the malaria infection to persist for a long time, increasing its chances of being picked up by another mosquito to continue its life cycle. Changing your appearance is one thing, but what if you could look exactly like the things your enemy is programmed to protect? This is the strategy of molecular mimicry. Instead of just being a new, unknown target, the parasite disguises itself as self. One of the most successful practitioners of this art is the schistosome, a parasitic flatworm that causes schistosomiasis. These worms can live for decades inside the bloodstream of a human host, a shockingly hostile environment. How? For one, they practice a form of camouflage called antigenic masking. They literally coat themselves with molecules they absorb from the host's blood, like serum proteins. They wrap themselves in a cloak made of you, making them invisible to your immune patrols. Beyond just borrowing a disguise, they can also manufacture their own. Schistosomes produce glycoproteins on their surface that are structurally almost identical to the host's own cellular markers. The immune system glances over them, seeing nothing foreign to attack. Some of their molecules can even mimic host hormones, potentially disrupting cell communication and dampening the immune response. The malaria parasite also has a few more mimicry tricks up its sleeve. On the surface of infected red blood cells, it displays proteins called rifins. These RIFINs have a very specific job. They bind to a receptor on our immune cells called LILRB1. In a healthy body, LILRB1 is part of a system that prevents autoimmune attacks. When it binds to a self molecule on a healthy cell, it sends a powerful "don't eat me" signal to the immune cell. By producing RIFINs that bind to this same receptor, the malaria parasite effectively hijacks this safety mechanism. It tricks the immune system into thinking the infected red blood cell is a healthy cell that should be left alone, allowing the parasite to replicate in peace. So far, we've seen parasites that hide and parasites that disguise themselves. But perhaps the most insidious strategy of all is active sabotage, directly suppressing and manipulating the host's immune system. These parasites don't just avoid the fight, they rig it from the inside. A prime example is Leishmania, a protozoan parasite that lives inside macrophages. Macrophages are the heavy infantry of the immune system, cells designed specifically to engulf and destroy invaders. But when a macrophage engulfs Leishmania, the parasite turns the tables. Once inside, it unleashes a chemical arsenal to disarm the very cell that's meant to be its prison and executioner. It blocks the macrophage from producing pro-inflammatory signals like interleukin-12, which are essential for calling in reinforcements and mounting a strong anti-parasite attack. Simultaneously, it tricks the macrophage into producing immunosuppressive signals like interleukin-10, which tell other immune cells to stand down. It's like a prisoner convincing the guard to not only let him stay, but to also tell all the other guards that everything is fine. Leishmania can also interfere with the macrophage's ability to show other immune cells what the invader looks like, a process called antigen presentation. By crippling this communication line, the broader adaptive immune response is never properly activated. Then there are the master manipulators, the parasitic worms, or helminths. These large multicellular organisms are renowned for their ability to create a state of chronic immunosuppression in their hosts. They achieve this by constantly secreting a complex cocktail of molecules known as excretory/secretory products, or ESPs. This cocktail is a powerful immunomodulator. It can skew the entire immune response. Your immune system has different modes of attack. A Th1 response is aggressive and good for fighting viruses and bacteria. A Th2 response is different, often associated with allergies and fighting off worms. Helminths push the immune system towards a Th2 type response, which is often less effective at killing them and can cause tissue damage that the parasite might even exploit. But more importantly, helminth molecules are incredibly potent at inducing the growth of regulatory T cells, or Tregs. Tregs are the peacekeepers of the immune system. Their job is to suppress other immune cells to prevent autoimmune disease. By stimulating a massive increase in Tregs, the helminth creates a tolerogenic environment, a state of truce where the immune system is actively held in check, allowing the worm to persist for years. These incredible strategies of disguise, mimicry, and sabotage aren't static. They are the result of a dynamic, multi-million-year-long co-evolutionary arms race. As hosts evolve new defenses, parasites are under immense selective pressure to evolve counterdefenses. It's a biological version of the Red Queen hypothesis from Through the Looking Glass, where you have to run as fast as you can just to stay in the same place. The parasite evolves to evade, the host evolves to recognize the evasion, and the cycle continues. This is why a huge portion of a parasite's genome is often dedicated to these immune evasion genes. The vast library of VSG genes in trypanosomes is a direct result of this arms race. On the other side, host populations also evolve. The classic example is the sickle cell trait in humans. In regions with high rates of malaria, having one copy of the sickle cell allele provides significant protection against the parasite. It comes at a cost, as 2 copies cause sickle cell disease, but the survival advantage against malaria was so strong that the gene became common in those populations. So what does all this mean for us? Understanding these evasion mechanisms is critical for human health. The ability of parasites to establish chronic infections leads to debilitating diseases that affect hundreds of millions of people worldwide. It also makes developing vaccines incredibly difficult. How do you vaccinate against an enemy that constantly changes its face? A vaccine targeting one of the malaria parasites' PFEMP1 proteins will be useless as soon as the parasite switches to another. However, by studying this biological warfare, we can find new weak points. Instead of just trying to kill the parasite directly, perhaps we can develop therapies that block its evasion mechanisms. Imagine a drug that prevents Leishmania from disarming macrophages, or one that stops helminths from inducing those suppressive Treg cells. This would allow our own immune systems to do what they do best: clear the infection. Ironically, the parasites that have become masters at manipulating our immune system may hold the keys to new treatments for our own diseases. By learning how helminths calm the immune system, we might develop novel therapies for autoimmune diseases like Crohn's disease or rheumatoid arthritis, where an overactive immune response is the problem. The silent war between host and parasite is a relentless dance of deception. Unraveling its secrets is not only crucial for fighting disease, but it also gives us a profound appreciation for the intricate and sometimes devious ingenuity of life itself.